So part of what we have yet to do, just as a reminder, is today we're going to be throwing out the idea of forming a society and seeing what you all think about that. And then tomorrow morning we will be meeting the different working groups. Will be bringing forth the recommendations that they'd like everybody to vote on. And then toward the end, there'll be the organizing committee will come together and talk to you about what we should be doing in the coming year about becoming a society. We're going to need to vote on that. And do we want to have a meeting? I would assume so. If so, where? Can we talk NIH into doing a freebie for us again? That'd be cool. Anyway, what? Oh, Tom's here. Okay, great. Okay, well, anyway, so think about tonight, about tomorrow. And is everybody set up to go to dinner that wants to go to dinner? Is there anybody that wants to go to dinner that hasn't RSVP'd? Okay, just a couple. I don't think they'll care. Okay, so Tom Kepler, a wonderful person and really a visionary scientist. He is a wonderful person. I'm not kidding. Anyway, he's going to be talking to us about
yeah, it's not the word, James.
That.
It's the hesitation. Again, I appreciate your willingness as well. One more time. Is the microphone not on? There it is. Okay, good. Thank you. So there's a huge amount of promise in the technology that we're here to discuss. And I'm going to focus my attention on one really small part of it. And I'm going to be directing my attention, again, to perhaps a relatively small number of people in the audience. But I think that it's these people who can really do a great deal. And I'm going to hope that we can inspire them to work towards some of these goals and with each other in ways that will help us to overcome some of these challenges. So one of the big promises, I think, that immune receptor repertoires provide is a window onto the history of antigenic exposures. At least in principle, every encounter between the immune system and antigenic microorganisms, whether they are pathogens or symbionts, results in a change, a permanent change to the immune system, which is then etched upon the receptor repertoire. So that, at least in principle, one could take this repertoire and, if one knew how, read back the history of these encounters. And I think that there's a great deal of potential in doing that. But I think that it's going to require a lot of clever thinking. So here's an example of a new technology that is potentially synergetic with the one that we're interested in. It was developed by Steve Elledge, and it is called the VIR-scan. And I won't go into the details except just to say that what Steve and his group can do is to take a drop of blood and test it on a panel of linear epitopes from human viruses and determine what viruses you have experienced in the past. So, this is sort of proof of principle that this information is in there. We have a much larger hurdle, however, to come up with that information using our technology, because we don't know what that map is. He's looking directly at what antibodies bind to which antigens and reading that off. We have to take an extra step from the sequences to the binding properties, and that's extremely difficult. Another example that points out a little bit more towards the power of the sequence information itself. This is a paper published by a collaborator of mine named Aaron Schmidt in Steve Harrison's lab, and what they did was they took the repertoire of an individual who was immunized for influenza recently, and went back and studied the patterns of mutation and determined that the clone that was elicited by the vaccine recently was originally elicited by an earlier strain and named the strain. And so he calls this viral archaeology, and I think it's just one really small example of the information that is locked away in the repertoire and that we potentially have the ability to reach. So the question really is, how do we get it? And there are a couple of different ways of thinking about it. The one that I favor is that what we ought to do is to understand the biological process that we're trying to get at, understand how that process encodes information into the repertoire, and then use that information to cleverly figure out how to decode that information and to estimate the biological process. Always the question is going to be, when do we believe it? When do we believe that the reconstruction, if you like, the estimations that we make about the biological process resemble in any important way the process that actually encoded the data in the first place? And that's really what this is going to be about. So there are two modeling approaches, and we've been talking about them. They have both been used during this conference, during this workshop, but we haven't really named them. And so I think it's pretty reasonable, I think, to draw attention to that distinction so that we can begin to recognize them and begin to work to put them together in effective ways. So the modeling approach that I grew up with is the generative model, although we certainly didn't call it that at the time. I only recently came to understand that it had a name. And this is a model that is based on a probabilistic process, a representation of what actually goes on in reality. And the whole idea there is that with a model like this, you can actually simulate data. You can take that model and generate simulated data. It's intended to be mechanistically complete to a certain extent. And it also gives you the chance to write down a likelihood function and do the appropriate statistical estimation and make inferences about the process using standard techniques from statistics. And this usually comes down to things like putting constraints into the model, and I'll illustrate that in just a minute. The other kind of model that is widely used, and perhaps more widely used in this community, is a discriminative model. Now, the discriminative model makes no attempt to actually get all the mechanistic details right. Instead, what it says is, let's look at all the features of the data, and we will learn what features are relevant to the question that we have in mind. This is the kind of model that's been used with great success in, for example, microarrays, which are used for diagnosis and prognosis of various cancers. The microarrays don't tell you what's going on in the patients, but it gives you, if you like, biomarkers that correspond, that correlate very well with the particular outcomes. And so that's another kind of approach. The difficulty that we have with each of these approaches, they each have their strengths and their weaknesses. The generative model, let's take the specific example of determining how to divide up a set of sequences into clones, or the simpler problem of deciding when two sequences are clonally related to each other. So in the generative model approach, which I've described before and Eric described to an extent yesterday, what you need is a model for VDJ recombination and a model for affinity maturation. And you put these two things together and you ask the question, do these two sequences look like they come from a single tree, a model in which there's a single tree, or do they look more like they come from a model in which there are two separate trees? So you've got a very specific model and you make a constraint. Is it one ancestor or two? On the other hand, you have a discriminative model, and you say, what are the features that help me decide whether or not these sequences are clonally related? And we already know what features we typically like to take out. Well, are they assigned to the same V gene? Are they assigned to the same J gene? And we have to say something about how similar the CDR3s are. So we have to make those judgments, and we'll decide based on whether or not these features in a given pair merit our describing them as related. And I'm afraid that I've skipped the slide that says what the downsides are, so I'll have to just remember them. For the generative model, the problem is that, well, first of all, the strength is that if you have the generative model right, it's going to be the most powerful method for inferring the truth, for inferring the underlying process. But that's no longer true if the model is wrong. And that's really the problem. So furthermore, we know the model is wrong, whatever model we're working with. So at some point, getting the model close enough is going to be good. It's going to do the job for us, but we don't know how close it has to be. And furthermore, there are important impediments to us getting the model right. And that's really what I'm going to focus my attention on in the next 10 minutes. In the discriminative model, where you've got, so what you would like to do is to be able to have a training set that says all of these pairs are related to each other and these are not. And what we'll do is we'll learn the features. Guess what? There is no such data set. And so on the one hand, we've got a problem that we understand the process pretty well, but there are details that matter and that we don't yet have in an appropriate mathematical model. And on the other hand, we have a method that works extremely well if only you had data. And so I think the point is that we really have to be aware of these shortcomings and begin to think hard about how we attack the problem by using both together to the extent that we can. So just for the sake of completeness, this is the way that a generative model would work. I have a phylogenetic tree, and I've got an ancestral sequence that emerges, that arises as a VDJ recombinant, and in the case that the two observed sequences are indeed clonally related, then something like this has to be the case. We don't know what the ancestor is. We don't know what the last common ancestor is. But what we can do is once we've got models that we trust, a model for how a particular sequence, how likely it is to have arisen by recombination, and the probability that starting with a particular sequence, we would end up with another one down the line. With all of these things, we can compute the likelihood, and we can compare it to the likelihood of a model in which the two are separate and make our judgments based on that. But as I say, these models are not complete, and we don't know whether they're good enough to make these inferences or not. So here, oh, there's my drawbacks. That was in exactly the wrong place. In any case, I've already gone with that. So what's wrong with the generative model? There are a couple of things. There are some major things, and Eric alluded to this yesterday, but I'm going to talk about just one of them. So we all know that the mutation rate is sensitively dependent on the nucleotides surrounding the nucleotide that is doing the mutating. This is the nature of the mutator enzyme, AID. And that's what I'm going to focus my attention on. Why that is still an outstanding problem. Beyond that, which I'm not even going to touch on, is the question of selection. Every one of the clones that we look at, with rare exception, have gone through selection as well. And that's another part of molecular evolution that molecular evolutionists themselves prefer not to have to deal with, because it is a very difficult question. In the end, we need to account appropriately for what we don't know. So I'm going to focus on item 1, local sequence specificity, why that is still an issue, and get it into the minds of the mathematically minded among you. We can estimate local sequence specificity. Many of us have done it, including the Yale group recently from AIRR data. What's the problem? Well, the problem is that just knowing what the sequence specificity is to start with is not sufficient, because it changes over time. When a hotspot mutates, it's no longer a hotspot. And you say, does that matter? And the answer is yes, it does. So I'm going to be looking at data from Joyce Wang in Fred Alt's lab. Fred has developed some very interesting mice that he has been able to use embryonic stem cells to insert any kind of a rearranged variable region gene, both heavy and light chain. And typically what he does is he puts in 2 of them, and this is a rag knockout mouse so that they don't make any of their own. He makes one, puts in one functional allele, so that keeps the B cells alive, and he puts in a non-functional allele. And it's, in this case, and the case that we're going to be using in our own work together, there is a stop code on the leader, which means that the passenger allele can experience mutations and it doesn't affect the fitness of the B cell. And so what we do is we study the pattern of mutations in the passenger allele. So the data that I'll be looking at is just one experiment in one mouse, and it'll be sufficient to make the point. It's just over 4,000 sequences, or nearly 5,000 sequences. They're all from germinal center B cells. And of course, they all share exactly the same unmutated ancestor. This is what the data look like, a fraction of the data in the alignment. And what I'm going to be doing is just counting up the number of mutations. And so this is a mutation in position 11 of sequence number 5. And so Y11,5 is 1 and is 0 in all of these other places. So that's all the data. It's just 1s and 0s in a big array. And so what I want to do is to estimate the mutation rate at each individual position. That's indeed what we're trying to do. What is the mutation rate at each position, and how does the local sequence context determine that? So what we're going to do is every individual sequence will have mutated for a lesser or greater period of time. And so that has to be accounted for. And so in addition to the local mutation rate, I have to also estimate the time over which each individual sequence has been mutating. And there's a fairly reasonable way of going about doing that. We'll write down an expression, and we're going to maximize this expression over mutation rates and the times. And I'm going to start with a really simple model, simply because I want to illustrate a few things, and the points that I'm making do not depend on the simplicity of this model. I can put in more complicated models up to a point and get the same take-home message. So we carry out that procedure, and we see this kind of plot. And if any of you who've looked at this kind of thing will recognize this immediately, we've got hotspots concentrated in CDR1, we've got CDR2, CDR3, and those are fairly recognizable, and even this new affiliated, if you like, CDR, which comes up in framework 3. I want to point out 3 different positions, because I'm going to be showing you a deeper analysis of these 3 positions. There's a hotspot at position 102, and adjacent hotspots at 314, 314, and 315. So they're adjacent nucleotides in a single hotspot. And what I want to do is look in more detail at the kinetics of the mutations at these positions. So here's the hotspot at position 102. Interestingly, it's not the canonical AGCT mutation hotspot. And here is a canonical AGCT hotspot. And both the G and the C in this motif are highly mutable, at least in this particular mouse. So here's the kind of thing I'm going to do. I'm going to take the position 102 hotspot, and I'm going to order my sequences by evolution time, and then I'm going to just count up cumulative mutation frequency. And then I'm going to see how well the model that I'm using fits to that. And so the important thing really is down here. This is where most of the data lie. And so the kinetics are not too surprising. They rise and then they flatten out a little bit, which is exactly the way the model predicts they will. But here's the interesting question. Suppose now I ask how many sequences have both mutations? I have got two hotspots now, position 102 and position 314, or position 314 and position 315. Okay? So in this case, the two hotspots are distant from each other. They're more than 200 bases apart. And these are the cumulative frequencies for the hotspots at these two positions. And this is the cumulative frequency for sequences that have both. Okay? Does that make sense? Okay. So in contrast, let's look at this one. These positions are adjacent to each other. This is the cumulative mutation frequency at these two positions. They both rise relatively rapidly. And this is cumulative distribution of those that have both. Is it clear what's going on here? What's going on here is that when one of these two positions mutates, it's no longer a hotspot. And so the mutation rate at the other position drops. So you see, if we were to start off by saying the mutation rate at this position is 0.79 and the next position is 0.82, it's not going to work. Or at least it's going to be an approximation that breaks down at some point as the clone evolves. At what point? I don't know. How much is that going to influence our inferences of selection, our inferences of clonal relatedness, our inferences of anything? I don't know. That's why it's an open problem. That's why I'm bringing it to your attention, because I think this is worth discussing. You can look at it a second way as well. It's the same comparison, only now what I'm doing is I'm looking at the same cumulative plots conditional on the mutation at 314. So this is the cumulative frequency of mutations at position 102 and at 315, unconditional. This is the same conditional on 314. That is, I'm only looking at the sequences that already have a mutation at 314. And again, you see a dramatic difference between the two. It's because one is adjacent, the other farther apart. Well, so there's good news and there's bad news. The good news is that mathematically speaking, we know how to solve this problem. We can write it down. In fact, this is the answer right here. The probability of finding sequence S after time t, if I've started with sequence A at time 0, is given by this expression. Voila, we're done. Except for one thing. This matrix M is 4 to the 360 by 4 to the 360. And so to compute this matrix exponential is going to be difficult. It's not difficult in the ordinary case, because ordinarily we treat each individual position as independent. Every column is independent, and so we don't have to exponentiate this gigantic matrix. This is truly astronomical. Instead, we exponentiate little tiny matrices, little 4x4s, and that's easy. And so what's really interesting and potentially frustrating is that mathematical solution is here. And so what you need to do is to find clever ways of finding an approximation. You've got the solution. How do you now find a way of computing this? How do you turn this into a computable algorithm? So the conclusion is, in my opinion, there's a great deal of information awaiting discovery. And there are really interesting and substantial mathematical challenges to be met. And I think that it's important to keep in mind the mathematical challenges as well as the computational challenges, and to try to bring them together to provide the kinds of analytical tools that will help us to get everything that we can out of these data, and indeed to really begin to combine the various approaches in clever ways. I just want to thank Fred and Joyce for allowing me to share this data with you, and of course the NIAID, who helped me along with my own research as well as this meeting.
So I would say I like the approach. I'm still a little confused about why sort of our models and other models that are based on sort of 5-mers would be incompatible with this, right? So basically, what we do is we take each base, look at the local sequence context, 2 base pairs up, 2 base pairs down, and then compute a mutability, a likelihood of that base getting targeted for mutation. But we would definitely say that you have to recompute that. So when you're analyzing the sequences, when you're doing these models, every time you introduce a mutation into the sequence, one then needs to completely recompute the probability of that base being hit. And so when you get that mutation at 314, that would actually reduce the mutability of the other position.
No, you're absolutely right. And what you're sort of eliding, you would say, oh, we have to just recompute the thing. That's precisely where the difficulty lies. So let's look at it a slightly different way. Suppose we only need trimers. So pentamers, I agree, are better than trimers, but just for the sake of argument, suppose we can capture the appropriate information in trimers, which means that in order to understand what the mutation rate is at a particular base, I have to understand when the base next to it changes. Okay? Well, in order to understand when the base next to it changes, I have to understand when the base next to that base changes. And see, the thing is that the influence spreads from this central position, and there's no principled way to cut that off and to decide at what point you need to stop and recompute something. I mean, I think that what you're suggesting is a very likely way forward. What I'm saying is that it's a long way away from being done.
Isn't it numerical versus analytic in a certain sense?
No, I don't think so. I mean,
so the formula you put up would be sort of a closed form solution that would take into account all downstream possibilities. If you wanted to, in other words, if you wanted to estimate the probability of a certain sequence, you say here's a germline. It's going to get 10 somatic hypermutations. What are the odds of the possible daughter sequences with 10 mutations? So what you put up there would sort of be a closed form to estimate the probabilities over all those, and like you said, it would be very hard. And I think what I hear what Steve's suggesting would be, just solve it numerically, plug in, do it, make the mutation, recalculate, and instead of having it analytically like you have, then just do it by brute calculation and saying, okay, so if I get the first mutation is here, then I recalculate and I can get the second mutation maybe here or here.
So if I can inspire you to actually try to do that and make it work, I will have succeeded beyond my wildest dreams.
Yeah, I was gonna say, if you give me the closed form, I'll compare.
Oh no, not even closed form. I mean, if there's a reasonable numerical approach to it, I think that would be also tremendous.
I mean, I just want to agree with Tom. I mean, it's not just a question of like you can come up with a finite approximation, and this is something that I'm quite interested in, but it's not just a question of like calculating it because you have this just sort of infinitely, not quite infinite, but very large chain of dependencies that just keeps on going on and on and on because you don't know. The point is, if you are starting in one state and moving to another state, you have to account for all the different possible chains of mutations that could have led from one place to another. And I'm not saying that there aren't ways of approximating this, but it's not just a purely computational problem.
Thank you.
So our next speaker is Dr. Nick Haining from the Dana-Farber Institute, and I don't know how to change these slides. Yeah. Jasmine, you? Okay. Good. I'm a Mac person, so yeah. It's done it. There you go. So he'll be talking to us about T-cell receptor diversification.
Jamie, thank you, and thank you also to Felix for the invitation to speak. I have to confess I'm not a card-carrying sequence person. I'm a T-cell biologist, and so what I want to talk to you about today is a project where we've started to look at TCR clonotype structure, but from a T-cell biologist's perspective primarily, to try and understand the relationship between the differentiation states of a T-cell and the sequence structure of the TCR that it expresses at a single cell level. So, my lab has been interested in understanding or studying the differentiation of CD8 T cells for some time, and understanding the regulatory programs that give rise to the differentiation of effector CD8 T cells from their naive precursors, followed by the development of memory and sometimes the development of T cell exhaustion in the setting of chronic viral infection. And we found it very useful to study this differentiation program by the analysis of genome-wide expression profiles. What I'm showing you here are gene expression profiles of samples of human CD8 T cells representing different differentiation states. In green here are naive CD8 T cells, or at least what we call naive CD8 T cells, sorted on the basis of CCR7 and CD45RA double positivity from a number of donors. These in colors here are tetramer sorted CD8 T cells specific for yellow fever virus from an individual who's been vaccinated with the 17D strain of the attenuated virus, or with individuals infected with either hepatitis C, here shown these HCV-specific CD8s, or HIV. And the projection I'm showing you here is simply a two-dimensional projection that's similar to principal components analysis that just illustrates the global differences in gene expression profile. So along this axis here, you can see a sort of a vector of genes that differentiates antigen-experienced cells from their naive precursors. Along this axis here, one might imagine a vector of genes that differentiates functional CD8 T cells elicited by the attenuated yellow fever virus vaccine from their exhausted counterparts. So we've certainly made a lot of interesting headway in understanding the differentiation programs that results in these different gene expression profiles from looking at bulk populations of T cells. But the problem, of course, is that each one of these dots here represents a gene expression profile of an aggregate of thousands of cells that, as anybody who's looked at a flow plot will tell you, differ a great deal, even within, say, a tetramer positive population for instance, yellow fever virus, for a different set of markers, and of course encompass a wide range of TCR clonotypes as well. So each one of these dots encompasses a large degree of heterogeneity at the level of the differentiation state or gene expression, as well as the clonotype. So to try and disentangle this heterogeneity of the populations, we've been interested in using single-cell RNA sequencing as a tool to study both the differentiation state of T cells and how it relates to the TCR clonotype. And the reason why is simply that we want to go from an average expression of a complex population of cells to individual cell identities, frequencies, and clonotypes. And the analogy that we use is that it's the difference between a smoothie or a fruit basket. So, conventionally what we've been studying all these years is a blended, homogenized mass of analytes from a large number of cells, whereas actually what we wanted to do is we wanted to individuate the individual types of berries here, and let's say I was particularly fond of Queen Anne cherries, which I am. It would be very hard to identify the relative contribution of Queen Anne cherries to the smoothie on the right. So no discussion. So this is the rationale then for trying to understand the relationship between T cell state and TCR clonotype at a single cell level. So no conversation about single cell RNA sequencing can proceed without a first quick mention of the technology that we're using, because it is a rapidly moving field, and the talk we give in 6 months' time may be based on an entirely different technology. In essence, single-cell RNA sequencing is currently being done in in two broadly different fashions: a sort of low throughput or a high throughput fashion. Low throughput or plate-based single-cell RNA sequencing is done on limited numbers of cells, in the sort of tens to hundreds of cells, that are isolated the way we immunologists love to do it, by individual flow sorting individual cells into a defined well in a multi-well plate, and by doing full-length RNA sequencing of amplified libraries from these individual cells. The alternative is to massively parallel single-cell RNA sequencing, which is to take vast numbers of cells in the sort of 2 to 10,000 range, isolated from a pool of cells. So you don't really have a control over which of a population of cells you're looking at, compartmentalized not in plastic plates, but in either an oil and water emulsion or in nanowells. And instead of looking at full-length RNA sequencing, based on the fact that one would need to look at tens of thousands of libraries, instead looking at 3' digital gene expression based on UMI barcodes, as we were using technology similar to that, which we've heard about already. For the purpose of today's talk, I'm just going to be talking about some data we've been generating using a low throughput. I think the challenge of combining genome-wide expression profiling and TCR clonotyping from massively parallel single-cell RNA sequencing is a significant task, and it's still a work in progress. The second aspect of the technology is obviously the analysis. Every lab that does this has their own unique blend of incomprehensible acronyms, and we are no different. We have the good fortune of working with a really smart guy called Nir Yosef, who runs a computational biology group at UC Berkeley, and together we've developed an analytic pipeline which I won't go through in detail, but basically uses a very clever iterative normalization process that he's developed called SCON. So sorry about these acronyms. Followed by clustering and projection T-SNE with MSC3. You get the picture. Anyway, so this is the analytic pipeline that we've used. I have to go through this in detail, but it's an approach that we've used principally to identify subclasses of single cells based on their gene expression profile. But in addition to what we've historically done in my group, which is to analyze the gene expression profiles of T cells, we've also been interested in finding out what else we can learn about each T cell. And obviously each T cell is particularly unique, like its own little snowflake, by virtue of the TCR sequence that it has, particularly the polymorphic CDR3 region illustrated here. Now, the simplest way to do it on any given cell would be to do it the way that one does for bulk populations of TCR sequencing, which is to sequence with read lengths that are long enough to span the CDR3 region, because obviously this doesn't map to a transcriptome, so you would need to have a read that's long enough to flank an annotated, say, V and J region in order to be able to read across the polymorphic region. However, it's not just because we're cheapskates, but actually it becomes sort of financially implausible to do long reads for large numbers of libraries. And so for gene expression profiling, we've been using short reads, 25 base pairs, paired end reads, which of course are not long enough to reach across the sort of average of whatever 40, 50 nucleotides that a CDR3 sequence represents. So instead, what we've done is to take an approach to develop an algorithm to reconstruct the CDR3 region from short reads in a process that, has another acronym, TRAPEZE, which is the TCR reconstruction algorithm for paired-end single-cell data. And this has really been developed by Shahed Afiq, who is a graduate student in Nir Yosef's lab. So this process starts by identifying, say, the V and J segments simply by aligning reads to known consensus sequences for V and J sequences. So that way, for each cell, you can define the V and J usage in the transcriptome. But in addition to these reads that align, we're also left, and remember these are paired end reads, we also end up with a large number of orphans. So rather than having reads that end up with a foot in both camps, or with pairs of reads that have a foot in both camps, you have a collection of reads for which one pair of the reads aligns to a known V or a J segment, but the other pair doesn't align. And so we collect all these orphan reads from pairs that have that align to known V or J segments, and use those to try and reconstruct what we think is the unknown CDR3 in an iterative process, where we take an orphan read that overlaps, say, partially with the V segment, and use that as an anchor to start reconstructing the unknown CDR3 region by establishing consensus nucleotide sequences from overlapping pairs, as shown here. So we've applied this reconstruction approach from paired-end short read single-cell sequencing data that has simultaneously been used to define the gene expression profile globally for thousands of transfer. Yellow? Yeah, by all means.
Just before we go too far, when you're trying to reconstruct the CDR3, there's going to be multiple receptors that use the same V and J combination. So how do you, are you reconstructing like one CDR3 for every?
From a single cell. You were, and full marks, because it is, you know, that postprandial nadir. Yeah, you know, you were definitely paying attention. So we've applied this TCR reconstruction algorithm to paired-end short-read data from single cells, and that has simultaneously been used to establish the gene expression profile of the cells for a number of data sets. And so, for instance, we have data sets from animals infected with LCMV, and we've been tracking the CD8 T cell responses at a single cell level, from individuals, from healthy individuals infected with CMV, and we have parallel measurement of naive CD8 T cells from those individuals. Yellow fever virus vaccinees, the naive T cells, and effector memory CD8s from those populations, as well as HCV specific T cells and their naive counterparts. From each of these different data sets, we isolated using single cell sorting somewhere between 15 and 130 single cells, and for a subset of these, we've profiled each library using 25 base pair paired end reads and 150 base pair single end reads to compare, which we're using essentially as the gold standard. And so for these, we've directly sequenced the CDR3 by looking at the sequence of a read that reads right across the CDR3 versus reconstruction using short reads using 3 algorithms: Tracer, a reconstruction algorithm that was recently published by Sarah Teichmann's group; Trinity, which is just your bog standard, you know, a de novo reconstruction algorithm for RNA sequencing data, and Trapeze. This graph shows the fraction of cells that have a productive TCR in which we were able to make which were able to identify the CDR3 or sequence CDR3. And for the top group of data sets, we have both 150 base pair direct reads as well as reconstructions. So these vertical lines represent, in essence, about as good as you can do if you directly sequence the CDR3. And the dots represent how well you can do relative to that gold standard using the algorithms. And as you can see, for these 2 mouse data sets, you get extremely good, you get as good a recovery of the CDR3 sequences using reconstruction as you do when you directly sequence with 150 base pairs. For these human data sets, it's not quite as good, but on average we get about 75% of the sensitivity, in the sense that if you can sequence it with 150 base pair reads, about 3/4 of the time you can reconstruct the TCR, and the specificity is almost 100%. So the agreement between our reconstructed CDR3 nucleotide sequence and the directly sequenced 150 base pair is almost 100%. And just I've also added in some other data where we don't have a gold standard 150 base pair, but you can see around about the same fraction of, we're able to recover CDR3 sequences from about the same fraction of T cells. The different populations that we've sequenced using this reconstruction approach have the expected clonotype diversity, which we've measured here in with a Gini index. The naive populations from each of the donors, shown here as a representative from one donor, have an extremely low Gini index, as you'd expect. Interestingly, the yellow fever virus specific response has a very heterogeneous response clonotypes as well. In contrast, the response from this healthy donor infected with CMV has a very high Gini index, and the vast majority of clones originated from a single a single parent. So how then does the TCR sequence data that we've generated using this reconstruction approach match to the global gene expression profiles of the individual T cells from which the TCRs are sequenced? And for this purpose, we've compared two CD8 T cell responses, one to CMV, a latent virus that causes a persistent or latent infection, together with naive CD8 T cell controls, or yellow fever virus, which is elicited by a single vaccination, a single encounter with antigen, together with naive and effector memory controls. We generated gene expression profiles assaying somewhere around 4,000 transcripts per cell in somewhere in between 50 to 100 cells per class, and use the clustering strategies that I showed you before to identify the major subtypes of groups of cells that clustered together. And we can see that from these sort of 5 groups of cells, we could identify 3 main clusters. Importantly, we compared how well naive CD8 T cells from these 2 different individuals clustered, because that would be an important control. If we had significant inter-individual variability, we would expect those naive T cells to be different. But reassuringly, in data that I'm not showing you here, the naive T cell profiles from these 2 individuals were essentially superimposable, meaning that I think it's reasonable to compare the gene expression profiles of T cells, of different epitope-specific T cells from these 2 individuals. So the single cell gene expression profiles that we see cluster in 3 main clusters. One cluster captures almost all of the CMV-specific CD8 T cells. A second cluster captures all of the effector memory CD8 T cells, which are CD45 RA negative, CCR7 negative cells, and these are the blue dots here. And the third cluster captures all of the naive CD8 T cells from both of the donors. But as you can tell, you don't need to be a computational biologist to realize that the yellow fever virus specific CD8 T cells essentially split in 2, a population in cluster 2 that looks very similar to effector memory CD8 T cells, and a population in cluster 3 that looks similar to, if not identical, to naive CD8 T cells. And to illustrate the kinds of differences in gene expression, here I'm showing the relative expression. So this is a sort of a combination between a sort of a heat map and a scatter plot, where the relative expression of transcripts for CCR7, LEF1, and KLRG1 are shown here, with red being a high level of expression. And this is, for reference, this is cluster 3 that contains both naive CD8 T cells and this naive-like yellow fever virus specific cells. And this cluster are uniformly high for naive markers like CCR7 and the transcription factor LEF1, but are almost uniformly low for a marker that's frequently found in antigen-experienced cells, KLRG1. This is true for not just these markers, but also for a large number of markers shown here by this heat map showing differential expression of now many more markers between CMV-specific T cells in cluster 1, effector memory, and a subset of the yellow fever virus-specific CD8 T cells in cluster 2, and naive and the naive-like yellow fever virus-specific CD8s in cluster 3. And as you can see, these naive-like cells show expression of many of the same markers that do naive to cells, but have very low levels of many of the markers that one might expect from an antigen-experienced cell, like granzymes or a BLIMP1 or NK-like receptors like KLRG1. So to further confirm that the expression profile of these naive-like CD8 T cells really did recapitulate a naive state, we turned to a collection of data sets that my lab has generated recently, where we've generated a set of almost 5,000 gene signatures compiled from published gene expression data sets, where we compared, for instance, naive CD8 T cells to their activated counterparts or dendritic cells before and after LPS stimulation to create canonical signatures corresponding to cell states and perturbations in the immune system. And together with Nir Yosef's group, developed an algorithm to look for the relative enrichment of each of these cell state signatures in single-cell gene expression profiling in an algorithm called FastProject. And this is the kind of output of the algorithm that we see here. So here for reference again is the clustering plot of each of these types of CD8 T cells that I showed you, and the relative enrichment of these signatures here. For instance, the signature of genes upregulated in effector versus memory CD8 T cells is represented by red in cells that have high aggregate expression of this signature of genes, and by blue in cells that have low aggregate expression of this signature of genes. And as you can see from effector signatures in the left two panels or naive signatures in the right two panels, all of the cells, whether they're naive or naive-like yellow fever virus-specific CD8 T cells, lack effector signatures and have significant upregulation relative to their non-naive-like counterparts of a full group of genes characteristic of naive CD8 T cells. So remember I mentioned that as immunologists we like flow sorting individual cells into plates. The advantage is that we do this on a fax arias. We can get 8 colors of flow cytometry data or whatever at the same time on the cells that go into individual plates. So we can actually compare the gene expression profile of individual sorted cells to their expression of a subset of proteins measured by flow cytometry. And this shows a kind of fax plot where we've quantify the relative abundance of CD45RA and CCR7 measured by flow cytometry on each of the cells that are colored according to their gene expression-based classification. So here are the effector memory CD8 T cells, here are the effector memory like yellow fever virus-specific T cells, and the open circles here are the naive-like yellow fever virus cells, and as you can see they cluster in a quadrant very similar to naive CD8 T cells as well, suggesting that the yellow fever virus-specific T cell compartment contains both cells with an effector memory phenotype and a naive-like phenotype. The question then, of course, is there anything in the TCR usage or structure that could explain the difference in this differentiation state of CD8 T cells elicited by a single antigenic exposure in a healthy individual that results in the generation of two T cell populations really quite markedly different gene expression profiles. So we surveyed a large list of attributes of the TCR that might be different. All of them were negative except for one, which was CDR3 length. So if we look at the CDR3 length of the effector memory CD8 T cells from this cluster, and that corresponds to these red lines in these cumulative distribution frequency, compared to the yellow fever virus-specific T cells with a naive-like phenotype, you can see that there's a significantly, that the distribution of CDR3 lengths in effector memory-like CD8, the yellow fever virus-specific T cells, is significantly shorter, both for alpha and beta, than in their naive-like counterparts. And that corresponds not just to the number of amino acids in the CDR3 lengths, but also to the number of additions or subtractions of nucleotides from the CDR3 itself, which you can summarize as a germline score. So the higher the germline score, the closer it is, the closer the sequence of the CDR3 is to the unadulterated regions sequences, whereas the lower the germline score, the more substitutions have occurred. And as you can see, the naive-like CD8 T cells have a significantly lower germline score, suggesting that they're much more divergent from the original germline sequence than their effector-like counterparts. We then asked, if you didn't know that there were these two subpopulations, but just looked instead at the CDR3 length of the entire population of yellow fever virus-specific CD8 T cells, what are the sort of gene signatures that tends to correlate with the length of the CDR3 region? So here I'm showing you on the x-axis the CDR3 length in amino acids, and on the y-axis an aggregate score for the enrichment of a naive signature, naive versus effector CD8 T cell signature. And as you can see, there's a significant positive correlation between the CDR3 length and the representation of a naive-like signature, or put the other way, there's a lack of that naive signature in T cells responding to the same epitope from the same viral challenge in T cells with a shorter CDR3 sequence. In fact, if we take all umpteen hundred gene expression signatures from our immune signatures database that correspond to CD8 T cells and ask which of those gene expression signatures correlate best with the length of the CDR3 sequence, for alpha and beta, you can see that the vast majority of significantly correlated are positively correlated gene expression signatures that tend to be upregulated in cells with a longer CDR3 sequence, tend to be those associated with a naive CD8 T cell signature. So what I've shown you is that this reconstruction algorithm called Trapeze allows efficient reconstruction of CDR3 regions relative to the gold standard of directly sequencing across it from short-read single-cell RNA sequencing data. And at the CDR3 region length in yellow fever virus-specific CD8 T cells, at least in the donor that we've looked at so far, appears to correlate with the differentiation state. Why would that be so? So what is the significance of having a longer or a shorter CDR3 region? Well, data from many groups, including this from Han and Robin's paper from some years ago, suggests that the distribution of CDR3 lengths in public versus private clonotypes is not the same. So in this paper, he sequenced the TCR repertoire from, I think, 7 or 8 different individuals quite deeply and identified the characteristics of CDR3 regions from individuals that were shared across individuals versus those that occurred only in one individual. And as you can see, those that are shared, shown in these red lines here, tend to be shorter, whereas those that are private to individuals or non-shared tend to be on the longer side. And this is the sort of observation that Danny and Vanessa Venturi and David Price hypothesized many years ago to be related to how easy it is to make a particular rearrangement of a CDR3 region, in that there's an uneven representation of CDR3s based on how much effort it takes to make it, in the sense that a shorter, a shorter, more germline CDR3 region is easier to make, and therefore occurs at a higher precursor frequency than a longer CDR3 region, which tends to occur less frequently, and is therefore less likely to fall into a particular antigen category of cells that could recognize through a particular antigen. So shorter cells, shorter CDR3 regions, easier to make, more abundant, and they tend to be the public clonotypes. Which leads to one putative model, although I will freely admit there are many others that would explain our data, which is that the CDR3 length governs the precursor frequency, which in turn helps shape the CD8 T cell differentiation state of T cells that are capable of responding to a particular peptide MHC combination. So in the naive T cell pool of cells capable of responding, for instance, to yellow fever virus, there will be those with shorter CDR3 lengths will exist at a higher precursor frequency, and by chance alone will encounter an antigen-bearing antigen-presenting cell earlier, faster, and more frequently, leading to earlier recruitment of those cells and more extensive differentiation. Whereas those that have a longer CDR3 sequence tend to exist in the pool at a lower frequency and may well end up entering the CD8 T cell response later. Whether this is a finding that we see just in yellow fever virus specific CD8 T cells, or whether this is a general phenomenon that actually helps account for some of the diversity of differentiation states that we see in the CD8 T cell compartment, is something that we're of course going to have to measure in more people and more tetramer positive cells from different infections. But I think based on this, strongly suggests to us at least, there's a very strong rationale for being able to simultaneously survey the nucleotide sequence of the T cell receptor and understand how it affects the differentiation state of the T cell that expresses it. And with this, I'll stop and acknowledge all the people that did the heavy lifting. Kathleen Yates is a superb scientist in my group with magic hands for doing difficult experiments. Kevin Bai is a terrific computational biologist in my group, and this has been a part of an ongoing collaboration with Nir Yosef's group, and this work has been particularly led by Shakeda Fique, a graduate student in his group. We have a lot of entertaining times working with Alex Shalek's group at MIT. Danny Dueck and Sam Darko have been terrific translators of TCR sequence data for the uninitiated, and of course this is part of a long-standing program project grant with Rafi Ahmed, and in particular Rama Akonde has been the postdoc working in his group. So with that, I'll stop and be delighted to take any questions. you want to ask your question again?
So have you tried looking at people who've been vaccinated more than once with yellow fever virus and seeing whether the naive CD8 population goes away?
It's great. It has to be said that we're not the only people to have seen naive phenotype CD8 T cells responding to yellow fever virus specific cells. Daniel Spicer group had a paper in Science Translational Medicine last year that showed a similar phenomenon. He had the luxury of tracking patients from 25 years out after infection, and the relative abundance of those naive-like CD8 T cells appeared constant. He had a very small number of people that have been vaccinated multiple times. Most people don't get vaccinated multiple times with yellow fever virus. So it's but of those that he saw, that he didn't see that you diluted out or you expanded up the effector memory like phenotype.
I'm not sure if I've got this right, but I'm attracted to the idea of precursor frequencies and how that affects the kinetics of the response. But so, do you have the data to look at the diversity of the receptors within the short and the long CDRs? Because wouldn't the model suggest that the long CDRs would have less diversity and the short CDRs would have
You mean just at a bulk level within the naive compartment? Is that what you mean?
No, well, no, at the single cell level within the yellow fever specific.
So within our population, there were no two TCRs that were the same, or of the sort of 50 or 100 cells that we looked at. So
if it was to scale up, would you expect you'd start to see what you might call the public clonotypes in the short and
yes.
the private?
Yeah, you would. Yes, if you could see, and gets back to the point where our technology was relatively low throughput, but as we transition to simultaneously profiling gene expression and TCR sequence from very large numbers of cells, it's that type of question that I think we'll be able to answer.
And so, and this might be very naive, but have you tried to sort out yellow fever specific cells from naive individuals? Because isn't the implication that those cells that you're looking at are cells that they may not have been activated? Because they're the ones that weren't selected because they're at the low precursor frequency, even though their specificity might be.
So you're correct that probably the killer experiment, which would be one that would be exquisitely difficult to do, would be to isolate a sufficiently large number of yellow fever virus specific T cells from within the naive compartment of unvaccinated individuals and profile the TCR sequence of those individual cells across thousands of cells, and then you would expect that the long CDR3 region cells should occur at a lower frequency than the high CDR3. So that's technically.
There's a mission.
It sounds a bit tricky. But the alternative would be simply to actually profile the bulk populations or do it at the bulk level, I suppose. But even then, I think you'd be limited by the number of yellow fever virus specific cells that you could capture from within the naive T cell population. It would be detectable, but exquisitely rare.
Thanks so much. Thanks so much for a great talk. The next speaker is Dr. Tania Bubela, and she is at the University of Alberta in Canada, and is at the School of Public Health there. And she's been part of the organization here, giving us really wonderful advice and input on governance. And anyway, so she'll be giving a talk here on governance in Biomedical Research Commons.
Well, thank you very much for the invitation to be here and to provide some input in some very exciting developments in your community. So, I think what we're all here for is not just doing bench research or research in our respective laboratories, but the goal is to try to get that research into the clinic for the benefit of patients. And so usually we think about this as sort of a translational pathway or translational pipeline. And unfortunately, while the science can be super, super interesting and really cool, there are insurmountable and mega barriers between step A and step Z. And a lot of those barriers come from people in my world, in the regulatory and the commercialization, the business, and the venture capital space. So in your world, we've done a global landscape. We're just preparing it for publication of cellular immunotherapy clinical trials. And so the clinical translation and where these things are occurring, they're occurring at a rapid rate, and clinical translation is in fact global. And the countries where there is the greatest expansion in the number of clinical trials for cancer immunotherapy in particular is in the United States and China. And so those are two countries with fairly distinct regulatory systems. So if we look at the involvement or the engagement of industry in this space, it is increasing over time. So the two are the light blue and the green. So the green is where you have a partnership in funding between mainly between NIH or other public sector funders and industry. And the light blue is industry sponsored clinical trials. So there is an expansion of interest in industry. But the dominant, at least in early stage clinical translation, the dominant players and actors are still academic institutions. And this is true right through from phase 1 and into phase 2. You don't start to really see significant industrial support for clinical translation of cellular immunotherapies until you start looking at phase 3 clinical trials. And that, I think, is a little bit of a difference now. We've really seen a shift in where venture capital is ready to put its money. Since 2007, 2008 crash, the biotech bubble has burst a little bit. This is probably one remaining bubble in the space of investment in biotechnology or biomedical research. But the bottom line is the bulk of the research is done all the way through phase 2 in academic centers. And so why is that important? Well, the other bubble bursting fact that I have here, and this is sort of one of my favorite papers by Hay et al. In Nature Biotechnology, and it's kind of the gloomy prognosis. And it goes to a little bit about the extent to which we value that early stage work. And the degree to which we value it and its potential to actually land with patients has a lot to do with our thinking and our philosophy about the extent to which we manage it or we're willing to share it. And so the really sad news is that even for small molecule drugs, which are way simpler than biologics or cell therapies, you still have 60% that fail in phase 2. And the probability in a lead indication of a drug making it all of the way through to regulatory approval from entering phase 1 clinical trials is 15.3%, and for all indications it's only about 10%. And at the end of the day, that's not the end of the picture. We're starting to see a retraction in that post-regulatory phase, where the big elephant in the room is how is anyone going to be able to afford any of these therapies? Because in most of the world, unlike the United States, we have public sector health systems. And even in the United States, a lot of your insurance is in fact much more closely resembles public sector insurance than you think. And so, you know, it's a long haul through clinical trials and into adoption. So when you look at the history of, even of CAR T cells, and the only thing I want you to take away from this is the research into, on the one side it's the clinical translational side, and on the other side it's all of the engineering and the tools and the technology and the expansion technologies that needed to process in parallel, that this is incredibly interdisciplinary work, And it takes a really long time. So from the 1980s right through to 2014, and you still don't have a regulatorily approved CAR T-cell therapy. Probably will happen soon, but these are really long clinical trajectories. And so if you were thinking that you were going to attach intellectual property in those early stages of development, your patent life has well and truly expired by the time you're getting down into clinical development. So what I'm hoping that I've made a little bit of a case for, besides hopefully not depressing you all too much at the end of your conference, is that early research, preclinical phase 1 and now into phase 2, run in academic centers, is still relatively pre-competitive. And the reason that we want to promote this sort of pre-competitive research by doing things like what we're discussing here in terms of sharing data and materials is that it raises the level of knowledge of all R&D actors. It doesn't limit appropriation of innovation that's closer to practical application when you start moving into phase 3, phase 4, and actual technologies to get things in large-scale production into patients. It avoids duplicative research, and that's critical because part of that big failure rate in phase 2 is because all of the large proportions of those trials are testing minor tweaks of the same molecules. So the FDA is sitting on a plethora of data that tells you it's unethical to try this in patients because we know it doesn't work. But because we've got all of that data behind a firewall, we can't share it with you. We can't tell you that there's almost no likelihood that your trial is going to be of benefit to the participants. So, and the other thing is that every time you have to negotiate access to data or resources, there's a transaction cost that's associated with it. And so that makes that knowledge and those materials sticky. And so if you can limit those transaction costs and make those transactions as fluid as possible, you can remove a lot of the crud at the early stage. Okay, and so this is the philosophy and the reasoning that's behind a lot of the things that are spearheaded here at the NIH in terms of all of the philosophy around the different kinds of commons that are emerging, not just in the genomic space, but in a whole range of other areas. And these commons are supported by infrastructure whose goal is to improve access for tools and techniques for successful clinical translation. So all the things that we've been talking about, databases, biorepositories, biobanks, centralized GMP, to facilitate things moving into clinical trials. And one of the biggest roles that I'll talk about a little bit less because I think it's been really coming through in the discussions in the last couple of days is this issue of promotion of standardized tools and reagents. So commons have a really, really important role in promoting those kinds of standardization and quality control activities. But I'll talk about that a little bit less. So as a social scientist, when I think about the commons, I think about it from the perspective of this amazing woman, the first woman to win the fake Nobel Prize in economics. And anyway, she's a behavioral economist, and she studied natural resource commons. And what she saw was that the commons that worked, that didn't get overexploited in the context of natural resources, were ones in which communities came together to form governance structures that enabled the fair use of those natural resources. So what she added to our understanding of commons is that these are shared and managed resources, but they're vulnerable to social dilemmas, to free riding, to cheating, and to bad behavior, right? So they do need some level of governance. Like you can't just leave them as a free-for-all. But knowledge commons or research commons are a little bit different from natural resource commons, and that's because my pulling out or utilization of this knowledge does not interfere with anyone else's utilization of the knowledge. Okay, so the main issue that confronts knowledge commons is underuse. So therefore, there's not a case to be made for funding or financing and sustainability. And the value is increased as more people use it. And not only that, but you incentivize recontribution of the additional knowledge gained through the use. So you pull it out, you create this network effect of pulling out data resources, adding value, and contributing that knowledge or that added value back into the commons. And the other thing about these things is rather than being very localized, they're globalized, and so that can come with its own governance challenge. So this is the slide that I always put up when I talk to communities that are thinking their way through sort of data and materials sharing. It's not the first time. So this is, the physics community are really the leaders in this. You can't do big physics experiments without shared infrastructure and without large teams. Oceanography, climate science, all of these communities have commons. And the main excuses that you get in terms of those social dilemmas for not sharing data and reagents is the fear of being scooped. It's my data, it's mine. It's too much time and effort. Others are going to use it inappropriately, so they're going to stick it into patients. The technology is too limited, it's just too hard. There's usually some legal terms not well understood that are thrown out there as excuses around privacy and around liability that aren't well thought through. There's some perceived commercial value and institutions in particular hanging on to that perceived commercial value, although in general they're getting a little bit better at that. So the biggest community that I worked with was the mouse community. So this is a mouse, an animal model community. And this was the case of two very large-scale international consortia. One was the International Knockout Mouse Consortium, which was a high-throughput enterprise that's global in scope to construct a knockout, at least at a level of providing a cell line or vectors for every gene in the mouse genome. And now the next consortia layered on top of that is the International Mouse Phenotyping Consortium, which has a standardized high-throughput phenotyping pipeline for adding that value-added phenotyping data to those knockouts. So we asked a question. This is just basically where all of the repositories and data centers and high-throughput centers are. So it really is a global enterprise. And so we asked the question about whether proprietization of these basic research tools that are, in part incentivized by, there was some mention of Bidol yesterday, by current laws and practices, whether they hinder the establishment of a mouse commons. And what we found that in the heyday of gene patenting, so these are mouse genes. These are about as useless and valueless a piece of intellectual property as you can possibly have. And what we found was that in the heyday of everybody jumping on the bandwagon of gene patenting was alive and well for mouse genes. And when I took that to the community, basically a room full of researchers like this, but at the in London at the Royal Society, they all looked at me like I was completely bonkers and said, but they can't possibly still be, no one's maintaining those patents even if we got them. So we went through and did the analysis, and yes, they had not been pruned. They were maintained, which means money. You have to pay every year for your patent. They were maintained for the full life of the valueless patent. Okay, so did it have any impact? No. What we basically found was that patent infringement was well and alive and good. So regardless of the fact that there were all of these patents out there, the research community, so these are the number of publications on the top 100 published on mouse genes, and the blue ones are patented, And I mean really patented. At the time, these patents were valid. and then publications on other genes. So basically, what you can also see is that there's this lamppost effect of everybody studying the same genes, they're all patented, and everyone's infringing quite merrily. There were some impacts a little bit on publications, but nothing too outstanding. The really impactful patents in terms of establishing, or the proprietization in terms of establishing the commons, though, were these broad spectrums methods patents. So these are all of the, this is prior to CRISPR-Cas9, so CRISPR-Cas9, you could add that to the mix now. But these are all of the patents that were involved that needed to be used, all the techniques that needed to be used in a high throughput pipeline. And this is no longer an individual lab that isn't worth suing. These are large, very well-funded, like the Sanger Institute, right, where most of the high-throughput pipeline was occurring. And what this does is that it does two things. First of all, there's a risk of being sued. And the second thing is it makes the ensuing resource less valuable to industry, because you cannot guarantee that the resource has been created without infringing on background intellectual property. And so it becomes more, industry is more loathsome to touch or to take out or to utilize the resource because they might get sued for patent infringement. Okay? So these are some of the risks and some of the issues with background IP in this space. All right, so then when we get into looking at transactions on how this worked, it really works a lot like that model that you were showing, all right? So you have, in 1 and 2 there, you have individual production labs that are making knockout mice or any other portion of this resource in their own labs. And there's a certain level of sharing that just goes on amongst labs, from lab 1 to lab 2. Then you have the high throughput production centers that are mandated to put all of the resources into the repositories with the accompanying data into the accompanying databases. Then in the center, you have all of those repositories, and sometimes you have individual researchers that are also depositing into repositories. So the repositories are both collecting, oftentimes they're set up to take data or to take resources from the high throughput massive projects, including things like the Human Genome Project or IKIMC or IMPC. But they also, like the Jackson Laboratory, will take submissions from or deposits from individual research labs. Okay? And everywhere you see an arrow, there's a legal agreement backing up that transaction. So when you have the repositories in the center, if you're going to have a mirrored resource and you're really going to turn this into a global commons, there has to be some agreement, like a consortium agreement, as amongst those repositories. Especially if you want to enforce or say all of the repositories, if they're receiving this kind of data or materials, they then are going to be distributing it for non-commercial use to academic research labs, but potentially also to commercial organizations or research entities. And so the input, so any of the deposit agreements, will spill over into the terms for the taking out, right? So the more complicated you make the terms of deposit, the more complicated you make the terms for being able to receive materials or from the repositories. So this is why you need to think very carefully about the level of complexity that you want to add on the left-hand side of the spectrum, the way you want to coordinate things amongst the repositories, because all of that is going to impact on how easily you're going to be able to then distribute the resources or enable users to access the data. Okay, so this brings us to carrots and sticks that have been raised a little bit along the way. So funding agencies and journals are the most logical places for some level of carrots and sticks. And the biggest carrot for most academics is in attribution and citation, right? Enforcement capacity is generally weak, and you will hear that from the funders. If the NIH, the staff will say we do not have the capacity to enforce a lot of the things that you're talking about here. So you need to beware. You can set norms or standards for the community or community behavior, but make those as simple as possible, because without enforcement they have no meat. Yeah? Okay, so very, very quickly, I'm just going to mention the Structural Genomics Consortium. I don't know how many of you are aware of this. But this is for proteins, for human proteins. And it started in 2004. It is a not-for-profit charitable organization incorporated in the United Kingdom. It has a 300-person strong team in 6 countries, and it has a completely open access policy. So it's promptly placed its results, its reagents, its know-how in the public domain, and it's a patent-free zone if you're involved. And the really interesting thing is it's a public-private partnership. So the pharmaceutical companies, and some of the biggest name pharmaceutical companies are here, are listed, pay to play. But they also agree not to proprietize anything that's put in the public domain by the Structural Genomics Consortium. So there's papers that I can share with you by Alan Edwards. And again, the goal here was to try to provide tools and reagents to the research community to try to bust us out of that mold of everybody, whether you're in academia or whether you've got commercial interests in your patenting, working on the same stuff. So if you increase or make probes available for the less studied protein structures, you will incentivize research in those areas. And so they have created a pre-competitive commons where they have tools and basic knowledge of the structures of their proteins, moving right through into discovery and exploration. So they provide probes and epitope data. All of it is pre-competitive. It's entirely free. But it doesn't preclude any company or any individual then moving into drug discovery and development to refine those compounds as they affect targets. And so they're keeping it open in that space up to sort of phase 1, phase 2, and then enabling an expansion of interest, reducing duplication in research, and enabling proprietization as you move into the more valuable end of clinical development. And they're partnering quite substantially with patient and disease foundations. So I've basically covered that. So in conclusion, there are a lot of examples of these kinds of new collaborative research models, and they're really gaining momentum. And what we're seeing emerge from ones that are quite successful are best practices in governance of pre-competitive research infrastructures, especially when you pay attention to building community norms, which involves a little bit of rules for IP management, incentive structures, and enforcement. And part of what you're going to need to build and what you're starting to do in a meeting like this, or a series of meetings in the working groups, is you're really building that community trust and enhancement of social capital, which I think as society will help you do. So that said, science is hard, but sharing shouldn't be harder. And lots of people and lots of funders have helped me along the way. And then I have a list of, if you're interested in any literature on this, I've got a whack load that I can happy to share. Thank you.
This is really, I think, it's great stuff, and I'm glad that you're able to bring it to us. Something that I'm still not getting, though, about the transition from the pre-competitive commons to the post-competitive. So I've got a particular compound or a structure, and all of the companies are free to develop it. So will they develop it? Have they been developing under this model, given that other companies might be doing it at the same time?
Well, if you look at what a lot of companies do, we have more Me Too drugs than we have anything else, right? So, I mean, it's a first come, first serve in terms of developing, right? So you leap onto the interesting compound first. You're going to be the first one that can take off with that. But you know, in the real world, it's not as if oftentimes we're splitting hairs between, you know, compound A and compound B at the best of times, right? But yeah, so the companies are free to take that information and to move it forward into development. And they have a bunch of case examples, particularly in the rare diseases world, where that has in fact happened. So they're very open to partnering with different organizations. Of course, GSK is in there with both feet because it's partly Wellcome Trust funded. One of the large centers is at Oxford. And so there are very close ties there. So it basically enables those pharmaceutical companies to, quite frankly, have access to the best structural protein guys in the world, right? So you know, it's kind of a win-win. It is a partnership. And Aled and Chas Buntra in Oxford, they've been, you know, those companies there were many fewer of them when it started. And that number has been expanding in number, which tells you the value. And they started just with the protein structures, and then they moved to identifying targets, and now they've moved to probes. But the probes are fundamentally still research tools, right? There's an endless line of medicinal chemistry that is required to take those anywhere into clinical development.
Just wondering, do you have some data on how much companies supported actually this platform, and how much data they provided to this database?
Yes, I do, and it's in my slides in the material that they presented to me, and I don't have it at my fingertips, so I will find me after and I'll tell you the exact number. But they are supporting it in cash, not just in kind.
Yeah, just wondering. So really, I think the number would be important to actually know if they support much.
Yeah.
And if that's the case, then
yeah.
If that's not the case, then I was wondering, what is the limitation? Why don't they do that? And
they do. They
overcome actually to get them contributing to this database.
So there's a number of different initiatives kind of like this. So the Innovative Medicines Initiative would be another one where my understanding of IMMI is that quite a bit of the support is also for small biotech companies, and there the investment on the industrial side is much more in kind than it is in cash. With the Structural Genomics Consortium, it is in cash, but I'm sorry, I don't have the number at my fingertips. So it's both in kind and in cash. So I recognize that, or everyone recognizes in this space, that pre-competitive is a very blurry line, especially when you're talking with small biotech companies. And it's probably a little bit clearer with pharma, but with small biotech, what is pre-competitive for me might be competitive for you. And I think that's some of the things that IMMI has been really, really struggling with. It certainly becomes a struggle when the companies you're talking about are actually in the tools space, in the tools development space, because then any of these kinds of commons are in fact competitive with a company that's predominant business model is to supply research reagents, for example. But with the IKMC and the IMPC, things like companies like Charles River, and Regeneron are actually part of the consortium. So they actually get some benefits out of it as well, both in terms of access to government research funds, but also they are utilized as repositories. The same in Europe. There was a French biotech company that had to be brought in to be the distributor for the resource for commercial enterprises because they purchased all of those background IP licenses so they could take on the responsibility of guaranteeing the resource as background IP, non-infringing, to then distribute to commercial entities. So there are creative ways to partner with industry the whole way through.
Okay, our last speaker in this session is Dr. Janice Reichert, who is the editor-in-chief of a journal called MABS, which has been really pretty successful and awesome. And also she's the executive director of the Antibody Society, and we'll be talking to you about societies, forming societies.
Good afternoon, everyone. First, I would really like to thank Jamie and Felix for inviting me to be here to talk to you. I will be describing the Antibody Society to you. My objective, as I understand it, is simply to explain the society and how, if you all are amenable to it, the AIRR community might be incorporated. So let me describe it. Bear in mind that the important part is to understand the structure of the society, and I will make this brief. I will try to fit within 15 minutes because I think a dialogue with you will be more instructive than me just standing here talking about it. Okay. So I started with the basics. I'm going to answer the who, what, where, when, and why. Then I will discuss the specific challenges and current status of the society. All right, let's start with who. And I do think of the society as a who. We are people, and we make up the society. It is technically a 501c6 organization, nonprofit. The numbers and letters there, the 501c6, refers to the IRS code that defines the organization. You do have to file with the IRS. If you bring in money and don't pay taxes on it, you better be telling the IRS why. You do have to have an official designation from the IRS to be a nonprofit. We do represent both individuals as well as organizations, and the only requirement is that you have some interest in antibody research and development. So far, we have one employee, that's me, and that's only as of January 1st, and I'll explain why in a few moments. The remainder of the people who are doing the behind-the-scene works, they are all volunteers. We do have a 12-member board of directors. That board has a president, we have a vice president, we have a treasurer of the society. Very importantly, we have a clerk/lawyer. To file with the IRS is very, very helpful to have all of the papers done by a lawyer. We have a brand spanking new website as of the beginning of the year, and we do have several people volunteering who are the editors of that website. What do we do? All right, you need to do something. We engage in a variety of activities. The underlying theme is these activities broadly benefit our members. We engage in education. I develop content around what's happening in the antibody sector. I go out, I give presentations, I talk about the society. This is all part of education. We are affiliated with 2 different journals, MABS as well as Protein Engineering Design and Selection. Heads we do an annual issue so that the society members contribute to that particular issue. That usually comes out in October. And we do a lot of encouraging discussion and collaboration between various organizations involved in antibody R&D, and this includes companies, academia, and government. We have 3 main initiatives that we pursue ourselves or we support. One of those is a response to the WHO's new definitions that apply to the international non-proprietary names that they give out. Hopefully everybody is aware that that is a function of the WHO to give these names. And they recently changed the way that the definition that they use to give the XI, the ZU, and the U nomenclature, and they're using IMGT database as the gold standard for comparison of sequences with the sequences that the companies are providing in order to get a name for their new therapeutic molecule. The new definitions have not met with much agreement by companies, so we are involved in the dialogue with WHO over how that situation might be improved. The next generation sequencing data that this meeting itself is supported by the Antibody Society. Jamie Scott and Felix here are members of the Antibody Society, Jamie being one of the board members. We are also involved in the establishment of standards for characterization and validation of antibody reagents, and society, and as well as board members Andrew Bradbury and Andreas Plichten are spearheading that effort, and we are doing that as a collaboration with the Global Biologics Standards Institute. We also, hopefully you are aware and maybe have participated in the past, we organize the Antibody Engineering and Therapeutics meeting that occurs in San Diego, usually San Diego in December. So quickly running through where and when. We are a virtual organization. We do not have an actual office. We incorporated in Massachusetts. That's another thing you need to do as a legal entity, as a nonprofit organization. You need to be incorporated in a state, pick a state, and you have to file with that state every year. You also have to file with the IRS every year, so you do have a tax return that you have to submit. We were founded in February of 2007, and one of the challenges that I'll get to in a few slides is that though we were founded in 2007, it has taken quite some time to accumulate the money required to actually hire someone to then pursue additional sponsors. So you have a little bit of a chicken and the egg issue to start out with. You don't have the money, you can't hire someone, and then you don't have someone to be soliciting sponsorship money. It goes a little around in a circle a little bit, but we finally broke out of that, and it was at a very good time, so I want to discuss with you the why, but this is really why antibody therapeutics are interesting and hot right now. I'll show you a little bit of data about that. I do want to emphasize that I'm talking about monoclonal antibodies, so these are recombinant antibody therapeutics. The numbers are a little bit larger than what you saw from the previous speaker, and I want to emphasize she was talking about cellular immunotherapies. So for me, I do track the antibodies as they enter clinical studies. I have someone at the FDA who tells me whether I'm warm or cold or hot relative to the number that I have accumulated so far. When I haven't found them all, she says, there might be a few more. Go dig some more until I get to about the right number, and then she says, yeah, that's about what we've got. So I know these numbers are accurate because FDA told me so. In 2011, we had an all-time high of 81. Prior to that, we had numbers that were in the 50 to 60 range. So you see there was a little bit of a drop-off in looking at the years 2012 to 2014, but that was actually the normal range previously. Last year, in 2015, we had yet another record year, an additional 20-odd more than the previous high. So we had over 200 new, novel, completely different, so this is a first in human study for over 200 antibodies in 2015. So that was a really remarkable amount of activity going on, as you might imagine, in the preclinical space to enable those to move into the clinic. At the other end of the spectrum, you have the antibodies that are in phase 3. I do an article series called Antibodies to Watch, so that phrase refers to articles that are published in MABS in 2010 starting through to 2016. So the Antibodies to Watch in 2016 documents 53 antibodies that are in phase 3 clinical studies. But as you can see, if you look backwards, when I started in 2010, that number was only 26. So the number of antibodies in phase 3 has dramatically increased over that short period of time. So what that has helped to do is really drive up the number of antibodies that are moving into regulatory review and onto the market. This shows the number of antibodies that were approved in either the US or the EU in these given years. In 1997, that was an all-time high. It's like, yes, 2 approved. But as you can see, we've gotten a little bit better. If you look, though, the trend through 2014 was for a record high and then usually a decrease. We had 4 in 1998, but it dropped off to 0 in 99. We had 6 in 2009, but that dropped off to 1 in 2010. In 2014, we met that record and had 6 again approved, but instead of a drop-off, you see an increase. In 2015, that went up to 9 new antibodies gaining their first approval in either the US or the EU. And in 16, I could list them out, but I won't do it for the sake of making this a shorter presentation. There's certainly likely to be 10 and potentially 11 antibodies gaining a first approval in the US or the EU. So I hope with these just a couple of slides I've been able to convince you that we are seeing a tremendous activity that is pushing more antibodies into the clinic, getting them through to the pivotal phase 3 studies, which are critical to get through to regulatory review and approval, but things are actually moving along pretty well. So this is a great time to be working in the antibody space. This is just an overall slide. Though I talked about 50, it was 53 when I wrote the paper. It's currently more like 56 antibodies at phase 3. That's backed up by over 400 antibodies that are either in phase 1 or phase 2. So there's a tremendous number of antibodies that are in clinical studies now. Whether it's good or bad, it's up to me to keep track of them all. All right, so what else is going on? Just again, very, very briefly, the name of the game working in antibody space now is enhancing the functionality of a standard antibody. This is done in a wide variety of ways. You can take a canonical antibody and engineer it in a variety of ways, either tinkering with the protein or tinkering with the glycosylation. You can make antibody drug conjugates, and there's specifically the drug is a small molecule drug. You can make bispecific antibodies, you can make other type of immunoconjugates. In this case, when people are talking about antibodies that are linked to a cytokine, for example, or a protein toxin, those are the immunoconjugates. You can take a fragment of an antibody and develop that as a therapeutic. It could be a single chain variable fragment, it could be an FAB, it could be a domain antibody. People are also developing mixtures, 2 or 3 antibodies developed as a mixture. You see this most frequently in the infectious disease area. And there are also completely alternative approaches that, again, I won't go into all of the specifics, but certainly one that you would be well aware of are their chimeric antigen receptors. So there are plenty of activity covering a wide variety of space in terms of enhancing functionality of these molecules. Just very briefly, I want to put a little pitch in for the perspective article written by Janine Sherman and Paul Perrin of GenMab. And here really the point though is just to show the variety of these molecules that are still in the preclinical space It takes on average somewhere between 8 and 9 years for an antibody to get through the clinical development process, so many of these newer molecules have not yet had time to penetrate into the clinical process. So phase 1, phase 2, phase 3, it will take time for these to move into the clinic and transition through the various phases. Right, now just to switch back to some description again of the antibody society and overall challenges that face any type of a nonprofit organization. First and foremost, of course, is financial support. You really need to be providing compelling reasons for individuals to have a membership or for corporations to provide you funding. The income really should be sufficient to fund at least one person full-time or almost full-time, and also to include some benefits. That person is critical to be the face of the society and also to be the person who does the outreach to membership, does outreach to the companies to gain the sponsorship. If you don't have that, then you've got a fairly moribund kind of organization. It is also important to have that person corral and steer and organize and manage all of the volunteer effort. Everybody has the best of intentions when they say they're going to do something, but you know, we had this phenomenon for many years where the board would get together in December and have all these great ideas for getting things done, and by February everybody had forgotten what they said they would do. So that's where I come in now. In December, this coming December, I'm really looking forward to making sure these people have done what they said they would do. But just to find volunteers that will contribute year-round is really quite difficult, and you need to be always thinking about providing them feedback and encouraging them, but providing some other types of incentives, for example, travel grants, this sort of thing, to attend meeting, exposures to their work, for example. That is one of the motivations maybe for being a website editor. You get your name out there as on the posts, for example. You need to be thinking about how to encourage people to continue to contribute and to feel like their contributions are valued. And just overall for the organization, you need to have initiatives. The organization has to justify its existence. It needs to get things done. And again, getting back to the critical nature of the employee or employees that you have, they need to be people who get things done. So the society right now, my main jobs are right here, substantially growing the membership. The membership is currently at around 520. What I want to emphasize is that is 520 people who have actually bothered to come to the website and put their name and address in there. The corporate sponsorships, essentially what's happening there is the company is buying a membership for every single one of their employees. But getting those employees to come to the website and put in their name and address is a different matter. I am aiming to chase people and have that number up to at least 850 by the end of the year. Part of growing that membership is growing the corporate sponsorship. I mean, not only to get the funding, but also to increase exposure of the society. We are focusing on the major pharma firms. We currently have Boehringer Ingelheim, Janssen, Genentech, as well as Roche, and 9 mid to small size firms. Genentech is very recent. I believe some people from Genentech were here. If you're not yet a member of the society, you have a free membership. Go to our website and sign up. But also to emphasize, it takes time. Genentech, in theory, or I guess conceptually, they agreed to fund us in January. We actually received the funds in June. This is why I've not yet chased Genentech people, because we only got their formal sponsorship in June. So you do have to allow for that time. You have to engage in a dialogue with these companies. Some will just say, okay, send me an invoice and I'll send you a check, but then others have very complicated processes for getting approval for use of their logos, this sort of thing. And the other thing that I'm doing is substantially increasing the society's profile. We have a new website. We're putting up posts. We do have a member-only section for information. And me, this is me, the executive director. I am going out, I am speaking to different organizations. I'm giving 12 different presentations at conferences in the US as well as in Europe and Cuba. And I have to say that one was a little bit of a boondoggle. I'm looking forward to going to Cuba. But the whole idea is just to get out, get in front of different organizations and talk about what we do, and try to get people interested in being members, to contribute. And what I say at the end of every presentation is, please join us. And I say that to you too. Think about becoming a subset of the Antibody Society. We would offer the structure to you, and then underneath that, pretty much you're welcome to do whatever you want. And we will now engage in a dialogue about that after I thank the organizations that have given us financial support.
So I think the organizing committee, or we can have questions first. Sure. Oh, absolutely. I mean, I thought this was going to turn into a discussion.
Well, okay. I mean, I don't mean to derail the more general discussion. How much of the income of the society comes from members, and how much from the corporate sponsors? Because I'm looking at 520 members, and I'm not seeing how that supports a full-time person.
Are you saying I probably work cheap? The corporate sponsors each pay anywhere between $25,000 to $5,000. The benefits scale with the number of employees. So we cannot ask small companies that have 5, 10 employees, we can't ask them to pay a huge amount. They would pay 5. The mid-sized companies would pay 10. The large companies would pay 25. Individual memberships, no, that's a tiny portion of what. We're not making a lot of money off of $100 for an individual membership, so we don't look at that as a large income stream. The income stream is really more from the corporate sponsorship, and that's why, as I say, the board in the past was not that great about actively pursuing corporate sponsorship. It actually took us a while to build up enough money and the courage to say, okay, let's spend some of that money to hire somebody to actually move forward. But, we've already substantially increased the corporate sponsorship, increased membership. I think we've got a long way to go. I think we can expand a lot more.
Thank you very much for coming and sharing the story here with us. So the thing that I'm a little confused about, and maybe some other people are interested in this as well, so I'll ask it, and maybe this is the elephant in the room, but what is, aside from being able to have a cool website and a cool logo and have members, what is the real positive benefit to us as a society, or I guess I'm using society in quotation marks here, as opposed to not doing anything and just kind of forming it grassroots without formalizing it the way you've done?
All right, I guess I get to sidestep that question because that's going to become part of the general discussion. One thing that I would just throw out is it provides the structure, so you become part of a legal entity that is a nonprofit that has already filed those documents with the IRS. We have a person who turns on the lights and answers the phones and the emails, that sort of thing. So some administrative support. Besides that, it does help to have that umbrella organization above you. In the case of interacting with the WHO specifically wants to deal with entities that represent an industry, that represent a body of people. They don't want to deal with loose affiliations. So in some cases, it does help to have that structure in place.
Given that I've been involved with large member groups in immunology, the American Academy of Allergy and Immunology, a smaller group, the Clinical Immunology Society, consortiums of groups, the FOCUS, or Federation of Clinical Immunology Societies. And the question I have is, at the beginning of this, if there's, let's say I'm an executive at Genentech and someone is asking me for $25,000, what is the expectation? What is the kind of like, what is owed Genentech to become a contributor to a startup society?
Good question. And I can say from the major pharmas, Boehringer, Janssen, Genentech, and Roche, you'll notice they aren't up there separately. They will be up there separately. This is another situation where they agreed, but we're still working through the process before we can actually formally put their logo up, but they are considered separate from Genentech. So far, Boehringer, for example, Janssen, they did nothing. They just said, okay, here's a check. There is no like quid pro quo. There's no they'll tell us this or that or tell us a position on anything. They basically just say, yeah, we support your mission and here's some money.
So part of the reason why the Antibody Society, I think, is able to do that is simply because they've had a meeting going for how many? 20?
30 years.
Yeah, it's getting on to 30 years. So the pre-existing meeting has been going on for a really, really long time, and the society just naturally grew out of it.
Maybe I can comment. I don't speak for Genentech, but because I'm with Genentech and I've had 10 meetings, it's the antibody engineering therapeutics meeting, so I can speak some benefit that I joined Genentech pretty recently, so these were just the first 2 meetings I went to last year and the year before. For one thing, Janice present a very nice antibody drug development preview. What you see, the number of drugs that's approved, just like one of the slides that she would show at those meetings, but with very in-depth, insightful predictions or updates of what's going on in the industry, what company is developing what drug, putting all these information together in a nice kind of always the last presentation of the meeting that we all look forward to. And my department chair, Paul Carter, one of the inventors for Herceptin, one of the very first Genentech drugs, also is very supportive of the society and the organization of the meetings. I think it's just a very nice venue for the antibody colleagues, for the field of drug development people to come together to talk about science, discuss science, learn what each other's doing. And in particular, last year's meeting, Paul and a few other colleagues, one of the 3 initiatives that Janice listed, who putting together this new, seemingly a little bit unreasonable criteria, how you would characterize your antibody as human or non-human or humanized, and it's based on a simple blast cutoff to the IMGT database, and then all of a sudden your humanized antibody has to be named as a chimeric antibody or non-humanized antibody, which throw the whole society, the whole industry world, very, very surprising move, and it's not good for anybody, or the patient, or the doctors, or the industry. And I specifically remember one comment from the discussion session is that we have never seen this group of people so united against one thing. and that was the WHO naming conventions. That just some comments.
Yeah, that's really what we're aiming to do, is to work on initiatives and to provide information that doesn't benefit just one company. It's something that one company can't really take on themselves. So the initiatives are really broader than what a single company would do. Some of the things that she was describing, of the information that I generate, that's business intelligence research. It's showing the landscape of the entire industry. Typically, individual companies don't really take the time to have somebody do that, and they certainly don't send that person out to talk about it in at various conferences, not to sort of broadly disseminate this information. So that's part of our educational mission, is to provide that kind of information, just to let people know what's going on in a broad sense within antibody research and development.
Yeah, I think so. This is just one model, but I have a slide somewhere I don't know how to use. If it was a Mac, I could do this. But I was wondering if maybe some of the organizing committee could come here and we could have a discussion about, for the last half hour, about whether we want to be a society, regardless of the antibody society, and what that would look like. Oh, sorry, whether we want to be a society and what that would look like. Yeah, I think that would be a good idea. Yeah, the organizing, yeah. I don't know how to make this go. Yeah, just everybody. Everybody just chairs. Yeah, sure. So I just made a single slide here of some of the things that maybe we could discuss, and some of which we could discuss tomorrow. But here's this: do we want to form an organization, maybe joining the Antibody Society, maybe joining some other society, or just working toward being our own society and, having our own governance structure, et cetera. So that's one thing to discuss. I think the first thing is about whether or not we want to organize into a society at all. And so do people have what the benefits of that would be?
Right, yeah, sorry. Would anyone know, could we call ourselves a society and not formally file a tax and become a you know what I mean? Do we have a year or so where we could say, yeah, we're the AIRR society, but not have a formal tax relationship or whatever that gives you,
Right. We can't collect money.
I'm going to throw in which jurisdiction do you want to be in? You don't necessarily have to be a U.S. organization.
Cayman Islands?
Sorry?
Good point.
Cuba. Yeah. Sorry, you said Cayman Islands. Yeah. So you don't necessarily need, no, I don't know enough about, so my understanding, when you have a society of a loose affiliation of members, the issue is that you retain, the board of directors or the directors of that society would retain independent individual liability for anything that goes wrong. So what you're doing when you're actually incorporating, even if it's as a nonprofit, and that would be the same thing as as in Canada, we have societies acts in each province, so it's same in the UK, it's as a charitable organization. You're creating a legal entity separate and apart from all of its constituent members, right? So yes, there are some issues around filings and tax implications, but without being that separate legal entity, you wouldn't be able to apply for example, funding that supports things like journals or other things. So you've got to be a little careful. The other little piece that I would always say is don't reinvent the wheel if you don't have to. And then the question that I would have for the Antibody Society is this meeting has been a lot about shared resources and data and materials and putting together policies and guidelines. Would that be supported philosophically by the Antibody Society? Because if you were to become a sub-branch or a subgroup within the Antibody Society, I would expect that you would have to get some layers of approvals from your governing board in the Antibody Society that they would actually support the promulgation development of those guidelines. So you might lose some independence there.
So I'll just use this thing and stand up. I've gone to the board and talked to them about this, and it was really just the fact that, you know, it took so many years, and seeing it happen kind of for the Antibody Society to form out of this annual meeting, and then for it to actually have some money and be able to do things. I mean, it's really been a long time. So I went to the board and said, well, there's this group that's forming, and there are things that they want to do, and it's a whole lot of work to organize it and look after it. And I don't know what our governance structure would be. I mean, those are all things we have to figure out. But the fact of the matter is, to keep going like this, it just can't be done by 2 or 3 people. It's really, you know, as kind of a part-side job almost. So this was the idea where I went to the board, and they basically said, well, if you want to do this, that's fine, and you can be your own thing within the structure of the society. So I don't think there is much blowback that would be coming about. As a matter of fact, I think it would be hugely welcomed, the whole sharing picture. Yeah.
Just to emphasize, you're looking at 1/6 of the board right here. Jamie and I are on the board. And I definitely agree that the board is looking to expand. I mean, we want to be as welcoming as possible. So, I mean, unless somebody could come up with some reasons that we're not thinking of right offhand, that there are legal implications to things that we have to consider, all this sort of stuff. The board isn't looking to impose restrictions. We want to, yeah, that's not what we do.
Isn't the principal barrier I can see is whether TCR people would be interested.
I talked to them about changing their name, actually. Well, I'll tell you why.
I say.
Exactly. Well, the people are kind of open to that. I mean, I think we'd have, if we wanted to do this, we would just have to like figure it out and go to them and negotiate with them and see how they took it. But the point is, for instance, the antibodies meeting this year, antibody engineering and therapeutics meeting this December, they're reaching out. They're going to have a couple of sessions on CARs, and I could easily, they're very excited about anything that gets rid of cancer and so forth. And let me also point out that it's about half and half industry people and academics. And I have to say, from my point of view, we need more industry people in this group. We don't, on any of the working groups, do we have anybody from industry? No. So you industry folks, get on in. Anyway, but my point is that that those are major stakeholders in this science, in this work. So I think it's a good thing. And truly, we have not seen, like for instance, in the Antibody Society, in the antibody meetings, nobody gives a crappy, I'm doing an infomercial for my company talk. If they do, they will be shunned and embarrassed. So it's just not the level of, I mean, it's a very open academic attitude is what I'm trying to say. It's really not about your company. It's about your science. And that's the thing that would bring us together, really, I think, more than anything else.
So I think many of us, one thing many of us can clearly agree on is we want to have more meetings like this. So just, I mean, is part of this decision, does this mean that this meeting would then get merged with the antibody meeting? And how would that work? So if we want to get, how much of the funding, the money that the Antibody Society gets goes towards supporting that meeting, and how would that be?
Okay, so the antibody meeting is actually put on by a company, and they do make money off of it. But we are totally in control of all of the sessions, right? So what happens is they do all the footwork, they do all the setup, they do all the whatever. And so our meeting wouldn't merge necessarily at all with their meeting. I mean, what I think might happen is that we may get some of the people on the meeting organizing committee, because they need bioinformaticists there. They need to bring in the T-cell stuff and all that. If they're going to do that and bring in adoptive cell therapy and et cetera, then it may be that some of our people, I guess I'd call it, would wind up on that meeting organizing committee. But we would work to have our own meeting, and possibly in the future, if you wanted to, you could go to IBC, that's the company that puts on the meeting, and say, put on our meeting, right? But they're going to charge more for registration, and how much is it for an academic? I think it's like $400 is the registration for an academic, typically. But they do all the work, which is kind of nice. So there's an organizing, which would be us, organizing the meeting, and then they do all the follow-up, they pay for the people to come, they do all of that, they write the literature, they put out like right now they're doing they've invited people for the December meeting now. They've organized us to get our act together to invite people, but like I got together with George, and George Giorgio, and he's co-organizing with me a session on, I've been doing, AIR stuff for, I don't know, the last N years, and we're doing it again this year. So they're different topics. Does that give you a picture of kind of how that would look?
I'm not sure she fully emphasized, though, that certainly you're welcome to have your own meeting anytime you want.
Yeah, exactly.
Again, don't think immediately, oh, they're going to tell us what to do, or they're going to not let us do that. This other thing. It's no, you come in, you're a subset, and do whatever you want. Yeah, that's fine. If you want to have your own meeting, that's fine. But just bear in mind, as I think Jamie was trying to point out, is there's a lot of administration, management. It's a lot of work to put on a meeting, and if you all are happy to contribute and make that happen every year, then that's fine too.
So, you want to go ahead?
Yeah.
So I've watched and been a part of the organization of this meeting, and I just have seen the tremendous amount of work that Jamie and Felix have put into it in particular, and the whole organizing committee for sure, but them two in particular. And to me, the most compelling reason to join the Antibody Society and form a committee is just that it seems that that's what they want.
What we're worried about is that we're retiring in 3 years. But not only that, we've got to figure out a mechanism to turn people over, have our own governance structure, build out these working groups, and have them evolve. We don't even have a governance structure, really. We said, oh, we need some working groups. We did it out of the last meeting. Some people said, okay, I'm willing to get the people together, and then whoever wants to be a member of the working group can be. Felix has an email list of about 600 people right now. Isn't it about 600 people? Yeah, 600 people that he emails out to. And by the way, if you have anybody that doesn't know about this and you want them to know, have them email Felix and he'll put them on the list. And so, I mean, it's just really, it's really very undifferentiated, let me put it that way. I mean, we really don't have anything in place except for the working groups, and we can like completely put this off and just go for a meeting next year. Maybe NIH, we can talk to John Warren and seeing if they could pay for her again. or not. We could do it another way, but it is just a gigantoid amount of work. And if you can get a meeting company to do it for you and you have to pay $150 more, $200 more to get in, I don't know if you guys want to do that, but it would really allow people to do the real work that needs to be done, which is the work of the working groups.
Yeah, I'm on the board of directors for a nonprofit, so I can appreciate the challenge of trying to get a group together and keep them motivated and keep that group going, and having the board directors follow up and so on. So my personal opinion is having an existing bylaws and structure is going to be a real boost if it's in the interest of this community to organize. I have some questions, though. There was the mention of this being a subset of antibody. Now, I don't know what that means. I'm wondering if the bylaws for the Antibody Society actually define what a subset is, or whether that would be something that would be added. Yeah, and then the, I mean, so if it has to be defined, if you could maybe try to clarify that a little bit. And then the second part is, and you have it on list there, but I think describe the governance structure of the Antibody Society. Elections of the board of directors, or how does that turn over? How would we as a group become part of that leadership structure, and are there ideas on how that would work?
I'll start with the basics. Really is that, of course, I can't actually concretely answer any of those questions because we've only just sort of started thinking about things. What really needs to happen is, it's kind of like a mating game. You have to discuss a little bit, see who's interested, and then depending on, really, I'd have to go back and look at the bylaws to see what they might say about such a thing. Whether there's some amendment that might needs to be done, but really what has to happen first is that there has to be interest shown. Yes, we want to do this. Then when we have that sort of at least general direction, then we would just explore these other options. Or explore the particular things that need to be done to make it happen. I think there's a way to make it happen. It's more a matter of what's the commitment and determination to actually get it done. Add something to that?
Yeah, I think right now all we can really say is that if the interest is there on the part of this group, then it will happen. And it will happen according to how this group wants it to happen, because I think everybody on the part of the board was just very interested in getting this type of scientist involved in the larger picture, I would say. And it's not simply about let's make T-cell therapeutics and let's make antibody therapeutics. And it's really about this larger immunological knowledge base that's being generated right now and translated into therapeutics, I would say.
So I've been to the Antibody Society meetings in San Diego, and I thought, I think one can, if we want to legally analyze what are the consequences of joining it, there's no way of doing that. I think it's much more important to have a match in spirit. And everybody I've met there, I think the kinds of things that people want to do in the society matches perfectly with what I've seen people do here. So I think there's hardly any downside. Also, what's the worst thing that could happen? Is the Antibody Society going to take over immune receptor repertoires and force us to do things we don't want? There's a very easy way to get around that, right? Everybody would just leave and say, no, we call ourselves something else. And we're not going to follow those standards, but make our own new ones. So I see very little risk and a lot of upsides. And absolutely, I would not, having tried that once, totally not want to do organizing a formal society ourselves. I think that would be a waste of energy and yeah. I mean, having tax status for our own non-profit, that's a lot of work. That
It would seem to me that you're just layering on a second layer of governance, and that it's not insurmountable. The only thing that would be challenging, I think, is if your articles of incorporation are not broad enough to be more than antibody research. And so, it depends on how loosely worded your purpose statements are. You might that expansion and a change in name would probably be your biggest headache. But adding in a layer where you would start to have chapters or sections is very normal for societies. And then you would just, in your governance structure, build in some representation from each of the chapters. And you might actually want to, when you're rethinking through the chapters, you might want to think what other chapters you would want to have within your already, sort of like interest groups, right? And you would have some representation there from the board, because you might already have membership where you'd be thinking about having a chapter for clinical trialists, for example. And it's just the maturation of an organization. And then as members choose to become members, they can check off membership in not just the society itself, but in the different the chapters that the society has to offer.
I've attended the Antibody Society meeting in San Diego several times, and one of the things that I should say that maybe should be appreciated is because there are so many companies there, and that so many of the research that we do is not really probably represented in many of those companies that have had a biochemistry, molecular biology, protein engineering background. And it's a great opportunity for us to promote our PhD students and postdocs to get jobs, because ultimately that's, I've had a number of companies after visiting there who have asked me to send my students to go work for them. None of them have graduated yet, so they can't do that yet. But still, I think that that would be something that's really important, because we all have a unique skill set in this community, but we don't have jobs for them among this community only.
Well, and let me just add that I think in the, I'm like a pop-up, boink. Anyway, that in fact, I guess in the years, I don't know, I've probably been doing this for like 12 years with the Antibody Society, or with the meeting. All of this was initiated, by the way, by Jim Houston, and I have to tell you this story because it just kills me. So he was one of the co-inventors of single-chain antibodies, and he has been supporting these meetings. He came up with them 30 years ago practically. And one year Felix and I went to the meeting and he was completely bald. And we thought, wow, what happened? Well, it turns out that what's keeping this man alive are therapeutic antibodies. So it's pretty amazing. I mean, I just have to tell that story because I think it just kills me that it's. Anyway, but the point is that in that field, what they're missing, I think, is the biology, the cell biology, and a lot of what people here are actually doing. We don't have bioinformaticists coming to this meeting hardly at all. They're kind of a minor, subset and so forth. So, but there's been tons of interest in repertoires, in repertoire analysis, in B-cell and now T-cell biology. So the point is the interest is there, but we haven't had the people. And so sure, if we wanted to, we could do, and I think probably what would come out of this is the annual meeting would have sessions would be much more along the lines of this kind of work, is my guess. And it would be much more integrated. But at the same time, we would still have to have our own meetings to be able to do the work that we need to do. And as a matter of fact, there may have to be some spin-off meetings. And by that I mean, maybe we need to have a meeting where we really get together a single workshop on working out I'm just totally spacing out. The genes, the inferring genes, the database stuff, bringing Marie Paul back into our fold. I don't know how that's going to happen. And so on. Well, we had a talk this afternoon about the next meeting. Where should it be? I think it should be in Montpellier. We'll come to her, right? That'll get her and get her going. She'll probably go on vacation. Anyway, but anyway, my point is that we should also think about some of these more intensive workshops that may need to happen, because the working groups, can the working group people talk about what it's like doing all this virtually, once a month meetings or whatever you guys were doing? How's that been for you? I mean, these guys have been incredible with all the progress they've made, and yet there's so much more to be done. Can you speak to that?
We usually spend the first 15 minutes trying to figure out how to work Skype.
We use GoToMeeting. It works fine.
No, I don't
Oh,
mean
we pay
that.
for
I mean the actual work you've been doing.
Yeah. The work, I mean, we've been meeting regularly, monthly. The virtual meetings have been great, but I mean, there's clearly a benefit to getting together physically every once in a while. We definitely need to continue that.
Could I just add one comment that if we do decide to join the Antibody Society, the British contingent will immediately have a vote afterwards to decide whether we remain or leave.
Oh, God. Oh, that hurts. That hurts.
I just want to remind people that tomorrow, I think from 12 to 1, we return to this idea of the future of the society, or the future of whatever we are, and do we want to do a society? I think that's 12 to 1 or 1 to 2 or something. But it's not like we have to decide right now. This is just bringing up the ideas.
Yeah. Oh, sorry. Go ahead. Go ahead.
So why would it even have to be a formal subgroup in the society? I mean, it sounds to me there were sort of 3 initiatives that were listed that the society is focused on, one of which is this. And can't we just sort of, you're a board member, can't we just ask for them to organize the conference and just get it involved? Is there a benefit to having a discrete subgroup versus just using the current structure?
I would think that you'd want to be more inclusive. Danny would feel excluded if you called it the Antibody Society.
No,
we have to negotiate some changes.
Right, that part, but does it have to then be with, once we expand, if the definition gets expanded, which it would clearly have to, is there a benefit then to having a subgroup, a formal subgroup within that that's specifically?
Well, I think that the critical point is the working groups, right? We're trying to define standards that we want people to adhere to, or encourage people to develop certain resources or do certain types of research. And so, I don't think it's the AIRR society or the AIRR community that's the important entity. I mean, the AIRR, you had to choose that acronym, Felix, huh? The community is important because we're all here and we're all thinking about these things together. But as Jamie has pointed out, most of the important stuff that's going on is going on in the working groups, and that's why part of what we need to do today and tomorrow is to really encourage more people to join the working groups and to be active throughout the year and not just 1 week a year. And so, I mean, I don't know, maybe we just need to add 3 or 4 or 5 committees under the Antibody Society umbrella, and we don't have to be, the AIRR subgroup of the Antibody Society, but the subgroup is important in so much as that's where we get the work done.
Could I just add to that, though, a bit? And that is, although it may not seem that way, there is an overarching big picture to the 3 working groups. Like they're not,
Of course, and
so that's why.
Also, if we're still having separate AIRR meetings as a subset of the Antibody Society, we would also have, a committee for that. So,
exactly.
Or a committee that's interfacing with the company that's organizing the meeting for that. So it's not, I'm answering Steve's question with both yes and no. Like, it's not important, I think, necessarily from an organizational perspective that there has to be a formal subgroup, but it is important from a practical perspective, and there does have to be a group of us that are doing the work, and both in the working groups and in the overall organizing. Yes, absolutely, there's a similar connection between the different working groups.
Just one quick comment. I don't know if other people in the room feel this way, but this is the first time that I think adaptive immune receptor sequencing has had a platform to share science as well, sharing toolkits, sharing platforms, and it's been incredibly valuable to me. I've learned a lot, and so maybe if we're able to continue that in terms of a scientific meeting as well as working groups and standards, that may also help accelerate the field just as much as the standards.
Yeah, I mean, I don't know. I would argue that even if we do take the Antibody Society as a parent organization that we maintain, I like Tania's word chapter. I mean, I really think we need that formal focus on AIRR, and whatever we do, we really have to do a lot more effort reaching out to the T-cell people and the cancer immunotherapy people, and I think keeping that some sort of formal designation of focus on AIRR is important.
Yeah, I agree. I think it's very important that we keep our own governance and our own sense of identity and have our own objectives that we want to achieve. Just in terms of the other things, I mean, it's been such a great meeting, and it's certainly, I mean, I was scouring around for ages to find a conference that focused on this kind of thing, and I asked Jamie, she said, well, come to this one. And so we really need to keep it going. And we've, at the same time, there are so many things that we've said we want to work on over the coming year. It's important that we focus on those things. So to answer some of the other points there, I think the more that we can join existing organizations, that we can take advantage of other people to take away some of the legwork on the conferences, the more we can get done.
I think that one of the most important things was, you presented that earlier, is this, not just the umbrella, but the leadership there is probably the most important thing, right? I mean, that's going to be somebody whose job it is to wake up every morning and go out and get these companies funding, or organize the next meeting, or at least head it up, make sure people are following up with that. And from this community's perspective, right now, it's you guys up there, and so one of you or a bunch of you is going to have to continue doing that if we don't leverage something that already exists. So I don't see much downside either. I would agree with whoever said that earlier.
One more question. In the Antibody Society, do you have a public outreach side as well? Because that was one of the other things that was being raised here. And do you have patient representation or patient organizations on your board, or is that something that you need to do? So it seems to me that the society is the perfect place to bring in that side of the community, not just the researchers in the industry, and to have that whole knowledge translation, public or patient engagement piece built into your organizational structure.
Well, it's after 5, and we have to leave, right? Yeah. So let me just point out that the dinner is at 6:30. We're going to meet in the lobby. Actually, we're going to meet at the metro station, and everybody get a ticket and just go down one stop. And so we'll just meet at maybe 6 in the lobby on the end toward the metro. Does that sound good? Because we need to be there at 6:30. And then tonight afterwards, we'll probably have a party in our room if anybody wants to go. 6:01. Yeah, you can walk there if you want. It's kind of a long walk.
30. Oh, dinner starts at 6:30. I also wanted to mention they have you can bring your luggage tomorrow, and they have a closet they can put it in, and it'll be securely watched all day. So I think that was what I wanted to say.
Oh, and a big thank you to the speakers. The speakers were awesome. Thank you.